Infectious diseases, preventive medicine and clinical species conferences. Mammal and avian spirochetal diseases. Members of the order Spiracatales are helical, motile, unicellular bacteria 0,1 to 3,0 times 5,0 to 250 microns in size. Spirochetes have periplasmic flagella, ranging from 2 to more than 100 per cell, which are encased in an outer sheath and are responsible for their typical snake-like motility. The lack of free flagella distinguishes the spirochetes from other motile bacteria. The spirochetes are propelled along by rotating on their longitudinal axis and by flexing motions which are elicited by the action of the sheathed flagella. The order Spiracatales contains two families, Leptospiraceae and Spiracetaceae. The genera Brachyspira, Borrelia and Leptospira contain species that are of veterinary importance. The members of the genus Brachyspira, formerly Serpulina, are usually found in the intestinal tract of animals. Brachyspira hypodysenteriae is responsible for swine dysentery and B. Pillicicoli for spirochetal diarrhea in pigs. Both diseases are found wherever pigs are farmed. Brachyspira alvinopoli is enteropathogenic of chickens, Borrelia spp, with the exception of B. Suilla, are usually transmitted by ticks or lice that have been infected with them. Borrelioses in animals occurs throughout the world and is caused by a number of Borrelia spp, which initiate diverse disease conditions in a variety of animal species. Borrelia theileri infection has been associated with anemia, lymph node enlargement and petechiation of the mucous membranes in horses, cattle and sheep, while Borrelia suilla has been isolated from necrogranulomatous skin lesions in pigs. Infection by both these species are rarely diagnosed. Borrelia burgdorferi has been incriminated as the cause of fever, lymphadenopathy, lymphocytic proliferative synovitis, arthralgia, recurrent lameness and encephalitis in horses, dogs and humans. It may also cause abortion in mares. Family Leptospiraceae is divided into three genera, Leptospira, Leptonema, and Turneriella, but only Leptospira contains pathogens. Leptospira is currently divided into 22 genome species, of which 10 are known to be pathogenic in animals and humans, and 5 species that show intermediate pathogenic characteristics. In total, more than 250 pathogenic serovars are recognized. Leptospira bacteria are transmitted by direct or indirect, through contaminated environments, contact with infected urine or tissues of infected carrier animals. A wide range of mammals including livestock and domestic animal species may act as carriers of Leptospira. Clinical manifestations include hemolytic crisis, chronic interstitial nephritis, mastitis, abortion and recurrent uveitis, periodic ophthalmia, in horses. Livestock may also act as a source of Leptospira infection for humans, as chronic urinary shedding may persist for months to years after clinical or subclinical infection. Swine dysentery produced by Brachyspira hypodysenteriae. Swine dysentery, in its typical acute disease manifestation, is characterized by dysentery and the development of acute diphtheritic to necrotic enteritis, with hemorrhage of the cecum and colon in weaned, growing, finishing and adult pigs. It results in temporary loss of condition, chronic wasting, or death. It is an important enteric disease in many countries. The disease was first described in 1921 by Whiting, in the USA, who were able to reproduce it in transmission experiments using the gut contents of affected animals, thus proving that it was transmissible. It was recorded in many countries of the world, but the etiological agent was unknown until 1971, when Taylor and Alexander described the isolation of a spirochete and the reproduction of the disease by feeding pure cultures of it to pigs. Shortly after that, Harris and the USA confirmed their findings and named the organism Treponema hypodysenteriae. Both the organism and the disease have been extensively studied since then. This knowledge has been applied to methods used to diagnose, treat and control the disease, but has not yet resulted in a universally applicable vaccine. The etiological agent of SD is Brachyspira hypodysenteriae, formerly Serpulina or Treponema hypodysenteriae, the more recently described B. Hampsonii from North America, and B. swanatina in Northern Europe. These species are included in a group of large oxygen-tolerant anaerobic spirochetes found primarily in the large intestines of pigs and other mammals and birds. Brachyspira hyodysenteriae is 6 to L0 microns in length, flexible and active when viewed by phase contrast microscopy, and stains readily with aniline dyes such as carbylfuchsin. The cell contains a protoplasmic cylinder 350 nanometers in diameter with pointed ends and 7 to 14 fibrils inserting at each end and is surrounded by an envelope. Brachyspira hampsonii and B. Swanatina are somewhat shorter in length and have 10 to 14, B. hampsonii, and 7 to 8, B. swanatina, flagella per cell end, respectively. The organisms can be grown on blood agar in atmospheres containing 5% carbon dioxide, and 95% hydrogen. They form colonies 1 mm in diameter surrounded by beta homolysis after 48 hours incubation. Descriptions of the epidemiology and pathogenesis in this chapter are based on studies of B. hyodysenteriae, and not on B. hampsonii and B. swanatina. Swine dysentery occurs worldwide, is common in many European countries, and has recently been described as a re-emerging disease in, for example, the USA. However, official national incidence figures are scarce. Increasing numbers of herds are being founded using SD-free breeding stock housed in isolation. Dysentery-free pigs are being used in feeding enterprises, and separate site weaning and rearing operations are all reducing the prevalence of the disease in many countries. Swine dysentery is transmitted to healthy, susceptible pigs by the ingestion of the feces of affected or carrier animals or of material contaminated with their feces. Fecal shedding of the organism occurs during the incubation period of the disease, usually 4 to 14 days, its clinical course, and for up to 90 days following clinical recovery. Farms on which an outbreak of the disease has occurred remain infected unless depopulation and disinfection or a whole herd treatment have been carried out. At least 2 electrophoretic types, ETs, or restriction endonuclease analysis, REA, types have been identified on individual farms, and B. hyodysenteriae appears to be a recombinant species with an epidemic population structure in which a few ETs have spread widely. Pigs of all ages are affected, although the peak prevalence is in weaned pigs of 6 to 12 weeks of age. The disease can be particularly severe in sows at farrowing or mid-lactation. It is usually introduced onto farms by the purchase of infected pigs, and spread slowly from pen to pen by means of drainage channels or movement of infected pigs, by pig-to-pig contact, or by infected boots and implements. The morbidity rate may reach 75% of all pigs on the farm, and mortality rates usually vary between 5 and 25%. Some immunity results following recovery from infection with SD, and recovered pigs rarely suffer relapses upon reinfection. Antibodies to B. hyodysenteriae have been identified in the sera, colonic secretions and colostrum of recovered animals. Brachyspira hyodysenteriae can persist in moist feces at 5 degrees Celsius for up to 40 days and in feces diluted 110 with tap water for up to 60 days. It can persist in slurry for at least 6 days and in soil for up to 41 days at -20 degrees Celsius, 18 days at 14 degrees Celsius, 2 days at 22 degrees Celsius and less than 5 hours at 37 degrees Celsius. Experimental transmission of the organism in infected soil to susceptible pigs is difficult to achieve. Drying or disinfectants such as formalin, phenols and hypochlorite at routine use dilutions eliminate the organism from contaminated pens within hours. Studies show that oxidizing disinfectants act within minutes. Mice, rats, mallard ducks, crows, flies and dogs may be carriers of the organism in the field. Experimental infection of day-old chicks confirms that the domestic fowl could be involved in the epidemiology. Brachyspira hyodysenteriae isolated from mice on infected farms has been shown to be pathogenic for pigs, and the organism can be maintained in mouse populations. Brachyspira hampsonii and B. swanatina have been found in migratory waterfowl such as mallard ducks and geese, and they may be involved in the spread of the infection. Pathogenesis. Infection with B. hyodysenteriae is by ingestion. The organism passes through the stomach and small intestine, protected from stomach acid by mucus in dysenteric feces in field cases, and reaches the large intestine. There, it invades the mucus and crypts of the mucosa within 2 hours of exposure. The organism colonizes the mucus, and moves in it by means of its flagella. Inactivation of the FLA and FLAB genes reduces motility. It is attracted to substances such as sialic acid, L-fucose, L-serine and blood in colonic mucus. Infection with B. Hypodysenteriae alters the mucus layer with enhanced mucin production and disorganization of the mucus layer. The bacteria multiply in the crypts and invade goblet and epithelial cells, damaging or disrupting them. Invasion through epithelial cell junctions may occur. Mature epithelial cells may be damaged by hemolysins and hemolysis-associated proteins, of which 7 have been identified, among them TLIA, LEB, TLIC and LIA. LIA encodes for an acyl carrier protein, ACP, for acylation of toxins which may be needed for B. Hypodysenteriae to display its hemolytic activity. An inflammatory response, associated with mast cell degranulation and production of interleukin develops. Inflammatory changes, superficial capillary dilatation, and rupture in the large intestine result in loss of tissue and blood into the lumen, as well as in failure of the colonic epithelium to reabsorb chloride and sodium ions, which give rise to diarrhea and feces, containing blood and mucus. Death is primarily due to dehydration, ion imbalances and toxemia. A phagocytic response occurs within 2 to 3 days of the onset of clinical disease, and specific serum antibodies can be demonstrated within 7 to 14 days of the onset of clinical signs and persist for up to 19 weeks. Serum antibodies may be related to protective immunity, as pigs that have recovered from SD seem to be protected from reinfection. IgG, IgA and IgM antibodies present in the lumen of the large intestine will reduce the rate of mucosa colonization. Furthermore, fecal IgA, but not serum IgG, was increased in pigs challenged with B. Hypodysenteriae 17 days post-infection. Glucose and lactate levels in serum are unaltered during the infection, but gluconeogenic amino acids are decreased. Serum neutrophils that increase during infection could contribute to edema and epithelial erosions. During recovery there is an increase in IL-10 and B. Hypodysenteriae-specific antibodies develop. Specific antigens include an outer membrane-associated lipoprotein SPA, and a membrane lipoprotein BMPB. Studies of B. Hypodysenteriae infections in gnotobiotic pigs indicate that the organism on its own is causing the disease. Lesions in conventional pigs, colonized by other bacteria such as Campylobacter coli, Bacteroides vulgaris, acetovibrio ethanogenins, Fusobacterium necrophorum and other spirochetes, which may aid in the development of the lesions. Only B. Hypodysenteriae, however, will initiate the disease. The disease may be exacerbated by the presence of fermentable food residues reaching the cecum and colon, and, although disputed, the severity of the clinical signs may be reduced experimentally by the use of totally digestible diets. Clinical signs. The first signs of the disease in a group of pigs include restlessness, twitching of the tail, abdominal discomfort, demonstrated by the animal kicking its flanks, hollowing of the flanks, slight reddening of the skin and slight inappetence. A transient fever of 40 to 46 degrees Celsius may occur, but usually disappears at the onset of the diarrhea, which persists throughout the course of the disease. Diarrhea may be the clinical sign first noted, particularly if affected animals are housed in pens fitted with solid floors, but the fluid feces are much more difficult to detect in those with slatted floors or containing bedding comprising deep straw. Blood, mucus and, later, pieces of necrotic material appear in the feces, which are yellowish at first but later become brownish-red, liquid in consistency and foul-smelling. When recovery begins after 7 to 14 days, the feces revert to the normal color and consistency but may contain large quantities of mucus. Affected pigs show a rapid loss of bodily condition characterized by prominent ribs and backbones, a thin appearance, sunken eyes and a hollowness of the flanks, resulting from loss of colonic contents. The coat may be rough, the tail limp and the perineum stained with feces, some of which may be mucoid or blood-stained. Affected pigs show a variable reduction in appetite but all continue to drink. The feed conversion rate and daily live weight gain may be permanently depressed. Permanently stunted pigs may pass khaki-colored liquid feces. In sows the reduced bodily condition may result in depression of reproductive performance. Sucking piglets as young as 3 weeks of age may develop classic signs of the disease, although the peak prevalence is in weaned pigs of 6 to 12 weeks of age. The disease can be particularly severe in sows at farrowing or mid-lactation. Production depression on an affected farm may result in an extension of the rearing time from birth to 100 kg weight of up to 30 days, as well as a 15% increase in farm costs, as a result of the decrease in productivity and treatment costs. The presence of passive or incomplete active immunity, a therapeutic and/or prophylactic treatment regimen of inadequate duration or level, and the use of antimicrobial growth promoters may all reduce the severity of the clinical signs and make them difficult to identify as being part of the SD syndrome. Other enteric diseases may occur concurrently and affect its clinical appearance or outcome. Animals with chronic disease, low rectal temperatures, less than 38 degrees Celsius, and chronic khaki-colored diarrhea should be euthanized on humane grounds. Lesions. Pigs that have died of SD are usually in poor bodily condition, and the carcass may appear pale and faintly cyanotic. The changes typical of SD, which may be less specific in animals that have died of chronic disease, are best observed in recently affected pigs killed specifically for postmortem examination. On opening the peritoneal cavity, it is frequently noticed that the mesenteric lymph nodes are pale and swollen, but are rarely congested. The large intestine is the only organ consistently affected, the lesion being, in acute cases, that of acute diphtheritic to necrotic typhlitis and colitis. It is usually flaccid and may be dark reddish-brown or congested and have a shiny surface due to edema of the sericea. Pale patches 1 to 2 mm in diameter may be seen under the serical surface. These are hyperplastic lymphoid diverticula. The contents of the large intestine are fluid, foul-smelling and contain varying amounts of mucus, undigested food and necrotic material, and are brownish-red in color. The appearance of the mucosa varies with the age of the lesion. In early cases it is swollen and congested, but subsequently becomes covered with blood-streaked mucus and patches of fibrin and diphtheritic material. There is an extensive layer of necrotic material in older lesions, which may become dislodged in patches to expose hemorrhagic mucosa. In some cases, the liver is swollen and friable and the gastric mucosa inflamed. The stomach contents may be fluid or contain fibrous material of dietary origin in straw-based systems. Microscopic lesions in acute cases are confined to the large intestinal mucosa, which is inflamed. Much of the mucosal epithelium is shed and the underlying capillaries are congested. An exudate comprising cellular debris, fibrin, bacteria and red blood cells covers the exposed tissue and remaining epithelium, and edema and an inflammatory cell infiltration into all parts of the intestinal wall are present. Spirochetes can be visualized in the crypts of the colonic mucosa in the earliest lesions by microscopic examination of silver-stained sections, and these may be identified as B. Hypodysentery by application of fluorescent antibody, peroxidase-linked antibody or by in situ hybridization using specific probes. Diagnosis. Swine dysentery is presumed, on the basis of the history of the outbreak indicating a slowly spreading enteric disease that follows the introduction of infected pigs, the clinical signs of blood and mucus in diarrheic feces of affected pigs, and the postmortem findings of mucohemorrhagic to necrotic typhlocholitis. The presence of spirochetes in gram-stained air-dried or wet smears of feces or of colonic lesions provides additional evidence. Samples should be submitted as fecal swabs in transport medium, and colon contents or portions of colonic mucosa in sterile containers. In countries where postal services are efficient, they may be submitted by post. Isolation of the organism may be attempted on selective media using spectinomycin blood agar containing 400 micrograms of spectinomycin per milliliter or media containing additional antibiotics, but care should be taken in assessing the results as weakly hemolytic spirochetes may also be isolated. At present 7 Brachyspira species are recognized in the colon of pigs, and fecal samples containing more than one species of Brachyspira are common. Therefore, care should be taken to obtain pure isolates. Hemolysis and isolation rates can be improved by slicing the agar or punching holes in it. Diagnosis may then proceed by use of biochemical tests and be confirmed by PCR-based assays. The exclusive use of PCR excludes minimal inhibitory concentration determinations. PCR is usually performed on cultures, but can also be applied directly on fecal samples, although with reduced sensitivity. In conclusion, checking for strong hemolysis is the only reliable method of detecting Brachyspira. Strains causing clinical signs of SD and isolation remains necessary for a correct diagnosis, and is also required for susceptibility testing of isolates. Serological tests such as microtiter agglutination and ELISA can be used on a herd basis to identify infected herds. Studies suggest that neither is 100% sensitive or specific, and that neither can identify individual infected animals. The main problem is that these tests may detect antibody to common antigens present in non-B. Hypodysenteriae spirochetes and statistical interpretation may be required. Herd monitoring can be carried out using rectal feces swabs or samples from recently weaned pigs for culture and/or PCR. Differential diagnosis. The presence of fresh blood and mucus in the diarrheic feces differentiates SD from most other enteric conditions. Salmonellosis is often associated with fever, the diarrheas of postweaning colobacillosis, salmonellosis, and colitis due to other spirochetes rarely contain thick mucus or frank blood, and transmissible gastroenteritis and epidemic diarrhea caused by coronaviruses occur in explosive outbreaks rather than spreading slowly from pen to pen. Fecal blood in gastric ulceration and proliferative hemorrhagic enteropathy is altered and blackish. At postmortem examination, the restriction of lesions to the large intestine rules out all but diarrhea due to other spirochetes, which is a milder disease or infection. Trichuris sus and esophagostomum infestations should be excluded. Swine dysentery may occur with other diseases and then be atypical. In these situations, and following inadequate prophylactic or therapeutic drug treatment, laboratory examination is required for confirmation of the presence of B. Hypodysenteriae. In an outbreak, individual severely affected animals should be treated by parenteral injection of a suitable therapeutic agent. This is particularly important if feed medication is to be used. Medication of groups of affected animals is best done by administering drugs in the drinking water, but may be given in the feed. The antimicrobials given below may not all be available in any one country for regulatory reasons. Pleuromutilins, tiamelin, valnemelin, are extremely effective in treatment of sensitive strains when given by injection, tiamelin, once a day for 3 days, in the drinking water, tiamelin, for 5 days, and in the feed, tiamelin, valnemelin, for 14 to 21 days. Lincomycin may be given by injection once a day for 3 days, included in the drinking water and/or in the feed for 21 days. Macrolides may be given by intramuscular injection twice a day, tylosin, for 3 days supplemented by medication in the drinking water, tylosin, tavolicin, for 7 to 10 days, or in the feed, tylosin, tavolicin, for 21 days. All animals in drainage contact should be treated at therapeutic rates at the same time and their pens disinfected. They should, if possible, be moved to clean pens after treatment. Prevention. The best way of prevention is to keep the herd closed. Extreme care should be taken regarding visitors, transport vehicles, rodents and other vectors, and there should be no purchase or recruitment of live animals. The disease may also be prevented by administering a course of medication at therapeutic levels just after weaning and mixing, but should not be used as a routine measure due to the risk of development of antimicrobial resistance. Groups of treated pigs should then be housed in cleaned, dried and disinfected accommodation, and thereafter be maintained in isolation until slaughter. Maintenance in all-in-all-out systems is very effective, and a rearing or finishing herd can be kept free from the disease even when the breeding stock are infected. No vaccines have as yet been registered for use worldwide. Killed whole cell vaccines made from one or more serotypes and enriched with envelope proteins have been used in the field. They appear to reduce the prevalence of disease, to improve the performance of pigs, and to reduce the number of therapeutic treatments required to control it, but do not prevent disease completely or prevent infection. Management. General hygiene reduces the spread of the disease within a unit. The use of all-in all-out husbandry, coupled with proper cleaning, disinfection and drying further helps to reduce the spread. Transmission from house to house can be reduced by disinfecting pathways and by using disinfectant foot dips and separate implements for each house. Slatted floors, solid partitions between pens, control of flies and rodents and lowering of slurry levels all reduce the spread of the disease. It is difficult, but not impossible, to control SD on earth floors or in earth runs. Drying and resting causes the organism to die out, and disinfection of wooden pen furniture or walls and of earth floors can help free wooden pens or paddocks from residual organisms and allow restocking with clean pigs. Water for temperature control should be used carefully in piggeries, as it is a common route for the transmission of the disease. Water used for sprays and pig pools should always come from a clean source, and liquor from slurry systems should never be used to flush gutters or cool pigs. Pig pools should be for one pen of animals only and should drain directly into the slurry system. Pools should be drained and disinfected between batches. Eradication. Eradication has been carried out in a number of ways. Depopulation and restocking. The sale or slaughter of the infected stock is followed by cleaning and disinfection of pig houses, disposal of manure, an empty period to allow drying. It is best done in hot dry weather, and rodent and fly control. The clean unit is then restocked with SD-free pigs, usually originally hysterectomy derived, from a breeding company, and the herd is maintained in isolation except for the purchase of disease-free breeding stock or the purchase of semen. Such herds have a good chance of remaining disease-free indefinitely, providing appropriate biosecurity measures are maintained regarding visitors, transport vehicles, and rodents/other vectors. Purchase/recruitment of live animals should be avoided even from officially declared SD-free stock. Depopulation may take place as a single event, where the pigs may be moved to other holdings for finishing so as to reduce the period that the farmer is without income. Treatment of the whole herd accompanied by disinfection and rodent control. A number of agents, tylosin, dimetridazole, ronidazole, lincomycin and pleuromutilin, have been shown to eliminate B. Hypodysenteriae. However, before treatment determinations of the SD-causing agent should be performed. Treatment at the recommended rate, extended to double the usual period for safety, of the whole herd with an agent to which sensitivity has been established, followed by cleaning, disinfection and the isolation of all farrowing sows and their litters until both have undergone full periods of treatment, eliminates the disease. Breeding stock should be purchased from disease-free herds. Partial eradication accompanied by control. Where hygiene and management are good, the breeding stock may be treated and the organism eliminated. Treated pigs are isolated from the contaminated remnant of the herd by a disinfectant barrier along drainage lines which is moved forward ahead of the clean piglets being born. Sufficient space for cleaning must be cleared by partial depopulation of the feeding herd, and clinical disease should be suppressed by prompt treatment or continuous medication. Such schemes are often cost-effective, the cost being recouped within 6 months, and reduce finishing periods by 7 to 21 days. Isolation and treatment of all incoming animals should be practiced. Isolation of the herd and the purchase of clean breeding stock is essential for continued success. Monitoring for freedom from swine dysentery. A number of programs have been devised to monitor the success of eradication campaigns as well as the continued freedom from infection status of pig breeding companies. Herds to be monitored should not be subjected to the use of treatment regimens that can suppress SD. Antimicrobial growth promoters should not be used. Farm staff should monitor the herd for evidence of clinical signs of SD and report all outbreaks of diarrhea for investigation, not merely those in which blood and mucus are present in the feces of affected animals. Regular veterinary inspections that include inspection of slatted pens in which dysenteric feces are difficult to identify and rectal fecal sampling followed by visual inspection confirms clinical freedom from infection. A regular program of monitoring the post-mortem findings of any dead pigs helps to detect early or mild cases. Avian spirochetosis by Borrelia anserina. Avian spirochetosis is an acute bacterial infection transmitted by tick to a wide range of birds. Clinical signs are highly variable and generally nonspecific. Diagnosis requires identifying the infective spirochete. Several antimicrobials are effective treatments. Control methods include elimination of infected ticks and vaccination. Etiology. The causative organism of avian spirochetosis, Borrelia anserina, is an actively motile spirochete, and consists of 5 to 8 loosely arranged coils. The genome of B. anserina is smaller than the one reported for other spirochetes. The bacterium harbors several plasmids. Cultivation in vitro is difficult and needs special substrates. Borrelia will grow on barber stone or Kelly medium but loses virulence after 12 passages. It can also be cultivated in embryonated duck or chick embryos, or in young ducks or chicks. Epidemiology. Spirochetosis is found in temperate or tropical regions, wherever the biological vectors are found. The disease or the bacteria are reported from numerous countries, including Pakistan, India, Iran, Brazil, the US, Algeria, Sudan, Ethiopia, Nigeria, and Tanzania. The most common vector is Argus persicus, the cosmopolitan fowl tick. However, other Argus spp, Asanchezi, transmit the bacteria as well. Many immunological and serological types of B. encerina have been demonstrated in various areas. Mild strains are common. However, in many tick-infested geographic areas, morbidity and mortality rates might approach 100% in exceptional cases. Recovery from one type confers solid immunity against the homologous types for more than 1 year but not against heterologous strains. Relapses, reported for some human Borrelia infections, are unknown in B. encerina infection of birds. Therefore, any reinfection can be attributed to a heterologous type. Generally, an infected Argus tick can transmit the bacteria at every feeding and maintains the infection throughout larval, nymphal, and adult stages. The ticks also transmit the infection transovarial, and the F larvae are infective. Ticks remain infected despite feeding on chicks hyperimmune to B. encerina or on chicks with high blood levels of chemotherapeutic agents effective against Borrelia. Other vectors, lice, mosquitoes, some species of ticks, and animate objects, can transmit the spirochete mechanically to a susceptible host whenever the piercing apparatus becomes contaminated with blood that contains Borrelia. Ingestion of bile-stained fecal droppings containing the spirochete, contamination of feed or water, and cannibalism during spirochetemia can result in infection. After the bite of an infected tick, the incubation period is from 3 to 12 days. Clinical signs of avian spirochetosis are highly variable, depending on the virulence of the spirochete and the age of the birds, with young birds being more affected. Clinical signs include listlessness, somnolence, moderate to marked shivering, and increased thirst. However, infection may remain subclinical. Ruffled feathers, anemia, and pale combs can also be noticed, a scan in appetence, which can lead to decreased weight. During the initial stages of the disease, affected birds usually develop a green or yellow diarrhea with increased urates. The course of the disease is 1 to 2 weeks. Egg production in layers or breeders may be decreased by 5 to 10%, with a higher number of small eggs. Lesions. The spleen is enlarged, with petechial or ecchymosis, hemorrhages, appearing dark or mottled. However, a contrasting situation may be observed in pheasants, in which the spleen is reported to be small and pale. Occasionally, the liver may be swollen and contain focal areas of necrosis. Kidneys may be enlarged and pale. A green, catarrhal enteritis is common. Specific agglutinins clump the spirochetes in successively larger clumps during the terminal stages of the disease. Agglutination lysis then begins to disintegrate these clumps, and spirochetal degradation products are liberated, which may result in fever. Death occurs most often 1 to 3 days after Borrelia disappear from the bloodstream. Diagnosis: dark field microscopy, blood smears, PCR assays. Diagnosis of avian spirochetosis depends on demonstration of Borrelia in the blood, either as actively motile during dark field microscopy, as stained spirochetes in Giemsa stained blood smears, or by PCR assay. In young birds, the Borrelia may reach vast numbers per oil immersion field and persist for several days. Older birds usually have low numbers of Borrelia that are detected only with difficulty or not at all and that persist for only 1 to 2 days. Increased numbers of immature RBCs are noticed due to the anemia. Silver staining can be used to demonstrate the bacteria in tissues. Agar gel diffusion and various serological tests have been described but are of questionable value because of the diverse serotypes that exist in some localities. Spirochetal antibodies are readily detected in yolks of eggs laid by infected hens. Control and treatment. Antimicrobials. Eliminating the tick vector. Immunization with bacterians prepared from local strains of B. Anserina. Several antimicrobial agents are effective against avian spirochetosis. The most widely used are penicillin derivatives, however, aminoglycosides, tetracyclines, and tylosin are also effective. Tiamelin is labeled for treatment of spirochetosis in poultry outside of the U.S. but is only approved for use in swine in the U.S. Although tiamelin is effective in control of spirochetosis in poultry, it may cause dysbiosis and disruptions in cholesterol metabolism. Antimicrobials can be completely effective if treatment begins when the number of Spirochetes per oil immersion field is low or moderate. However, if large numbers of Spirochetes are present in the bloodstream, the sudden liberation of large quantities of spirochetal degradation products can result in higher mortality rates than with no treatment. Control must be directed against the biological vector. Argus ticks are notable for their long lifespan, ability to survive for extended periods without a blood meal, efficiency in transmitting the spirochete, and ability to remain securely hidden in cracks and crevices often beyond the effective reach of pesticides. Accordingly, control is difficult. A combination of tick eradication and immunization is the most effective means of control. Immunization can be highly successful and, next to eradication of the biological vector, is the preferred method of control. Bacterians prepared from local strains of Borrelia have been used for prophylaxis. Vaccines may be prepared from formalin or phenol inactivated material from lysates of blood, tissues, embryos, or eggs infected with B. anserina, and they may be lyophilized or liquid. Whole egg propagated bacterians are usually administered in 1 or 2 IM injections. Little if any cross protection is afforded to different serotypes. Birds normally have protective immunity after recovering from natural infection. Lyme disease in livestock, Borrelia burgdorferi. Lyme disease, or Lyme borreliosis, was first described in 1977 as a distinctive entity in a cluster of children from Lyme, Connecticut, USA. These children had symptoms resembling juvenile rheumatoid arthritis, a disease that does not cluster and is rare in children. Based upon similarities with recognized human clinical syndromes in Europe that were of unknown etiology, but associated with the bite of Ixodes ricinus ticks, the causative agent was isolated from North American Ixodes scapularis ticks in 1981, and subsequently named Borrelia burgdorferi. Once the connection was made between vector, agent, and the human disease, Lyme disease has been diagnosed throughout the world, but its significance is greatest in North America and Europe. Borrelia burgdorferi has become an important public health problem and the most prevalent tick-borne disease in the United States. It emerges across much of the northern hemisphere, causing considerable morbidity and in some cases mortality in humans, domestic animals, and occasionally wildlife. The wide geographic distribution and the broad host range of both the vector and the bacterium provide ample opportunity for livestock species to be infected with B. burgdorferi. Most of what is known about Lyme disease, however, is based upon clinical studies in humans, and experimental studies in laboratory rodents. Etiology. Borrelia burgdorferi is a gram-negative spiral or corkscrew-shaped diderm bacterium, measuring 10 to 30 microns in length and 0,2 to 0,3 microns in diameter, with a lipoprotein-rich outer cell membrane and periplasmic flagella. For culture in vitro, this microaerophilic slow-growing organism requires a complex liquid medium, and an optimal temperature of 33 to 35 degrees Celsius. The genome of B. burgdorferi is small but complex, composed of one large, highly conserved linear chromosome and many small, highly variable linear and circular plasmids. Based on 16S ribosomal DNA gene sequences, the genus Borrelia belongs to the order Spiracatales, family Spirocheaceae, but genomically unique and not closely related to any other bacteria, including the other spirochetes. Other related genera include Spirocheta, Christispira, Treponema, and Brachyspira. It was first believed that Borrelia burgdorferi was the only bacterium that could cause Lyme disease, but differences in morphology among isolates from diverse geographic locations suggested that additional spirochetes may be involved in the etiology. Borrelia burgdorferi is genetically diverse, and is represented by more than 20 different genospecies grouped within the Borrelia burgdorferi sensulato complex. Borrelia burgdorferi sensu stricto is present in the USA and Europe, but not in Eurasia and Asia. B. Phthalii, B. Guerinii, B. Balaisiana, B. Spielmanii, and B. Lusitanii are present in Eurasia. B. Japonica, B. Turdae, and B. Tanakii in Japan, and B. Bizetii and B. Andersoni in the USA. The causative genospecies of Lyme disease in humans and livestock are B. burgdorferi, B. Phthalii, and B. Guerinii. B. Balaisiana has also been proposed as a causative agent of the human disease. Borrelia burgdorferi belongs to a guild of pathogens, including Ehrlichia and Babesia spp. And tick-borne encephalitis viruses, that are maintained within the same vector reservoir niches. Thus, co-infection with one or more of these agents can occur and may be responsible for paralyme disease syndromes. Since these other agents have a wide host range, they also infect livestock. Epidemiology. Borrelia burgdorferi S.L. is geographically distributed in temperate regions of the northern hemispheres. These areas have the environmental conditions and reservoir hosts conducive to ixoid ticks. The range of B. burgdorferi S.L. was previously limited to northern Africa at its most southern extreme. Nevertheless, migratory birds, particularly seabirds, likely contributed to the expansion of its geographic distribution, with documentation of B. burgdorferi S.L. DNA in ixoid ticks or seropositivity in livestock, as far south as the Falkland Islands, Crozet Islands, and offshore islands of New Zealand, and in Chile, Brazil, and Uruguay. Borrelia burgdorferi and its vector ticks tend to be non-selective in their host range, and B. burgdorferi has been isolated from a wide variety of birds and mammals. Most hosts harbor subclinical infections. For example, in hyperendemic areas such as New York and Connecticut states in the USA, a high percentage of dogs are seropositive, and probably infected, but only a small fraction manifest clinical signs. A hallmark of Lyme borreliosis is persistent infection, even without antimicrobial agents, which is the rule in its many hosts. This has been experimentally proven in paramyscus mice, laboratory mice, rats, hamsters, gerbils, guinea pigs, dogs, and non-human primates, and in reported confirmed spontaneous cases in humans based on culture, and PCR. Among a large number of hard tick species, B. burgdorferi has been primarily detected in Ixodes ricinus, which is prevalent in Europe, Ixodes persulcatus, which is prevalent in Eastern Europe and Asia, and Ixodes scapularis and Ixodes pacificus in Northern America. Clinical cases of Lyme disease could therefore be expected in localities where these Ixodes ticks are common. During their life cycle, which could take from 2 to 6 years to complete, ticks feed as larvae on small rodent reservoir hosts, thereby acquiring infection. After the larvae molt and harden, infected nymphs attach and feed on a variety of birds, reptiles and mammals, including livestock. Finally, adult I. scapularis, I. ricinus, and I. persulcatus ticks seek and parasitize large mammals, including deer or livestock, and are considered to be the most common vectors of Lyme disease for large animals. Borrelia burgdorferi S.L. has been identified in other ixoid ticks that parasitize livestock, such as Haemaphysalis spp, Hyaloma spp, Amblyoma spp, and Rhipicephalus spp. Serosurveys for B. burgdorferi antibody in horses have revealed that the endemic and seasonal distribution of exposure closely corresponds to the prevalence of B. burgdorferi infection in humans in the same regions. Thus, like dogs within endemic areas, horses frequently seroconvert, indicating exposure and probable infection, but most do not manifest clinical signs. Lyme disease in cattle often occurs as a herd problem with a geographic distribution similar to that among horses. Pathogenesis. Transmission by the vector, nymphal or adult ticks, to the host is a complex three-way interaction between the tick, the host, and the pathogen. Hosts mount local inflammatory, hemostatic and immune responses to the feeding tick, while tick saliva contains substances that counter the host response, thereby facilitating a successful blood meal and transmission of the pathogens. After tick attachment, B. Burgdorferi spirochetes undergo striking variations in the expression of antigens, and migrate from the tick midget to its salivary glands. After being transmitted, spirochetes remain and multiply in the skin at the attachment site for several days, and then disseminate to many organs and tissues throughout the host. Motility plays a major role in the spread of spirochetes from the site of tick attachment, and allow contact with the host blood vessels. As a result of spirochete multiplication at the tick attachment site, the host reacts with a localized skin rash, erythema migrans, which generally persists for weeks after detachment of the tick. The rash is observed in the majority of human cases, and has been reported occasionally in livestock. Clinical signs of illness are generally most apparent during the early stages of disseminated infection when spirochetes are believed to elaborate pro-inflammatory lipoproteins that appear to facilitate the process of dissemination through host tissues. Despite the highly immunogenic nature of these lipoproteins, spirochetes effectively evade host immune clearance by yet-to-be-understood mechanisms. It should be emphasized that Lyme borreliosis in untreated humans, and experimental animals, is ephemeral, with spontaneous resolution, without antimicrobial treatment, of erythema migrans, myocarditis, arthritis, and other signs. Studies in animal models have shown that resolution of arthritis and myocarditis is mediated by the acquired humoral immune response of the host. Under these conditions, anatomically defined inflammation resolves, but infection persists. Whatever the mechanism, it is clear that spirochetes undergo dramatic shifts in protein expression. For example, outer surface protein A, OSPA, is a highly immunogenic lipoprotein that is expressed in the midget of ticks, but is rapidly downregulated upon onset of feeding, and is minimally expressed in the mammalian host. In contrast, OSPA is upregulated under similar circumstances. Up and down regulation have also been documented for several other lipoproteins, some of which are expressed exclusively in vivo, but their role is still unclear. Other possible explanations of immune evasion by B. Burgdorferi spirochetes have been proposed. These include sequestration in extracellular matrix, intracellular localization, formation of cystic-like forms found in vivo and in vitro, and the existence of complement-resistant strains that regulate and control complement activation. Once spirochetes have disseminated, some animals and humans develop clinical signs of Lyme disease, including fever, lethargy, weight loss, stiffness, swollen joints, laminitis, lameness, abortion, uveitis and, sometimes, encephalitis. In livestock, clinical signs have been observed in cattle, horses, and sheep. The mechanisms by which B. Burgdorferi spirochetes cause disease in infected hosts are being studied in animal models. Borrelia burgdorferi spirochetes produce no known toxins yet are capable of invading virtually any mammalian tissue and causing infection and disease manifestations for months to years. It is suspected that the approximately 150 lipoproteins that are encoded by the B. Burgdorferi genome will play an important role in not only disease pathogenesis, but also host immunity, and are considered to be virulence determinants. An important first step in the establishment of infection is adhesion of spirochetes to host cells and extracellular matrix components. Differential expression of pro-inflammatory lipoproteins, of which the majority are outer surface proteins, in various tissues and at different times during persistent infection appear to be critical determinants of disease. The genome has 12 linear and 9 circular plasmids, and loss of plasmids has been correlated with decreased infectivity and pathogenicity. Clinical signs. The incubation period for Lyme disease in livestock has not been determined. Observed clinical signs in naturally infected horses and cows correlated within a month of the seasonal emergence of adult ticks, indicating a long incubation period. No significant clinical signs were observed in experimentally infected cattle with cultured B. Burgdorferi, nor in ponies that were exposed to ticks. Experimental infection of a pony with pulverized B. Burgdorferi infected ticks resulted in seroconversion on day 9, which continued for a 3-week period, then waned. Rechallenge with cultured B. Burgdorferi 6 months later led to seroconversion within 4 days. Infection efficiency has been found to depend upon the infectious dose, the strain of Borrelia, and the age of the host. Experimental infection of cattle with different doses and different strains of cultured organisms demonstrated that the mean infectious dose was 10^6 to 10^7 organisms, with some variations in infectivity among different strains. Clinical signs in horses and cattle, when present, are varied and often vague and nonspecific. A skin rash resembling erythema migrans in humans is not a prominent feature of Lyme borreliosis in livestock, though there are rare reports of erythema rashes in cattle and horses at the site of tick attachment, which can be under the lower jaw, on the abdomen and ventral chest, on the legs, in the region of the mane of horses, on the udder of cows, and on the tail. In horses, clinical signs may include depression, low-grade fever, 38,2 to 38.8 degrees Celsius, swollen joints, shifting lameness, muscle tenderness, hyperesthesia, ataxia, behavioral changes and uveitis. Animals with disseminated disease can develop single or multiple swollen joints, arthritis, laminitis, stiffness, hyperesthesia, and sporadic lameness, which are the most common clinical signs. Untreated cases can persist for years with chronic weight loss, neurologic dysfunction and polyarthritis. In pregnant mares, there is a suggested association with borreliosis and reproductive failure due to fetal resorption or abortion. In cattle, stiffness, swollen joints, and/or lameness are the most commonly reported clinical signs, although reproductive failure, reduced milk production, fever and chronic weight loss are also observed. Arthritis has been most notable in the carpal metacarpal, tibiotarsal and/or stifle joints. Other clinical signs in cattle include abortion and decreased milk production. In small ruminants, clinical signs are not often observed or reported. Lameness, poor condition and poor appetite have been reported in seropositive lambs, but other studies have found that all seropositive sheep were healthy at the time of serum sampling. Sheep experimentally exposed by tick bite did not develop systemic infection. In the few reports involving goats, a high prevalence of B. burgdorferi antibodies have been found, but clinical signs were not evaluated. It has been suggested that goats will develop clinical signs similar to those in cattle. Lesions. It appears that Lyme disease in livestock is not fatal, signs are often ephemeral, and disease is mild, if present at all, so the pathology of disease in these animals is not well characterized. Post-mortem examination and histopathologic finding associated with Lyme borreliosis in livestock in general include arthritis, synovitis, and myocarditis. In spontaneous infections in horses, suppurative uveitis, necrotizing to non-suppurative meningitis and radiculoneuritis and non-suppurative myocarditis with intralesional argyrophilic spirochetes have been documented. In cases of uveitis, spirochetes have been visualized within the intravitreal suppurative exudate using silver stain. The neurologic manifestation of Lyme borreliosis in the horse recapitulates the human disease, and the equid is the only non-human host to develop neurologic disease after natural infection or after a biologically relevant route of experimental infection is used. A cow diagnosed with Lyme disease showed distension of the tibiotarsal joints. The synovial fluid collected from the joints was copious, watery, clear and amber. Endometritis and a few abnormalities in the abdomen were also found. In experimentally infected equids, lymphohistiocytic dermatitis at the tick attachment site with reactive regional lymphadenopathy were the only lesions identified. Although descriptions of abortion and metritis have been included in some reports, reproductive tract and fetal lesions have not been characterized, and the direct role of B. burgdorferi is as yet undetermined, but transplacental transmission of spirochetes has been documented. Diagnosis. The clinical signs of Lyme disease in domestic animals are very diverse, making an accurate clinical diagnosis difficult. An awareness of the tick vectors of the disease in the region and the exposure to such ticks are important for diagnosis. For seroepidemiological studies, assays such as immunofluorescence assay, enzyme-linked immunosorbent assay and immunoblot are commonly used. The gold standard for the diagnosis of Lyme disease, however, is the isolation of the organism from blood, skin biopsy, milk, vitreous aqueous humor, and synovial or cerebrospinal fluid. Culture requirements are stringent, as this microaerophilic, slow-growing organism requires a complex liquid medium and incubation at 33 to 35 degrees Celsius. A valuable ancillary procedure is PCR, especially real-time quantitative PCR, which detects spirochetal DNA, whether or not the organisms are alive. This is especially valuable when joint fluid, which tends not to contain viable organisms, is analyzed. Direct cytologic examination with identification of spirochetes in vitreous humor has been successful in cases of uveitis. Differential diagnosis of Lyme disease must include, among numerous other possibilities, traumatically induced carpal hygromas and chronic carpal and hock swellings, interdigital dermatitis, white line disease, rabies, spinal compression, brucellosis, leptospirosis, equine protozoal myeloencephalitis, and vitamin E or selenium deficiency. In horses, negative serology and normal CSF or joint fluid analysis does not exclude the diagnosis. For equine neuroborreliosis, histopathology is considered the most reliable diagnostic test. Control. Chemotherapy of Lyme disease in animals is based upon that used for the treatment of the disease in humans. Antimicrobials such as doxycycline, amoxicillin, penicillin, ceftriaxone and cefotaxime are effective against B. burgdorferi. Early treatment is desirable, as treatment is most effective when given during the early stages of infection. Chronic cases require prolonged treatment, which is often less successful. A bacterian and a recombinant vaccine based on OSPA are available for veterinary use, but there is no evidence of their efficacy in livestock. The best way of controlling Lyme borreliosis in livestock is tick control. This can be achieved by biological control of ticks, by application of organophosphates or pyrethroids in the tick's habitat, or by the use of ectoparasitic substances applied directly on animals. Leptospirosis in livestock, Leptospira spp. Leptospirosis in livestock is caused by spirochetes of the genus Leptospira and is characterized by fever, renal, hepatic failure and reproductive failure. In equids, leptospirosis may manifest as recurrent uveitis associated with an autoimmune response. Infection in livestock also may result in chronic renal infection and urinary shedding that persists for months to years following initial infection. Contact with livestock is an important risk factor for human leptospirosis, a widespread but neglected zoonotic disease that is estimated to affect more than 1 million people and result in around 59,000 deaths worldwide each year. Leptospirosis was first recognized in humans in 1886 by Weil, who described it as an infectious disease characterized by icterus. The responsible organism was simultaneously isolated and reported in 1916 by two independent groups of researchers, and shortly afterwards rats were shown to be a host of infection. Leptospira spp. are difficult organisms to culture, which presents challenges for the diagnosis of infection and has limited research into human and animal disease. However, the advent of molecular typing and, more recently, whole genome sequencing, has improved our knowledge of Leptospira taxonomy, virulence and transmission. Etiology. Leptospira spp. Bacteria belong to the family Leptospiraceae. Leptospira spp. are thin, 0.15 microns, right-handed helical, motile organisms that are 10 to 20 microns long with distinctive hooked ends. They are gram-negative organisms with an inner membrane and peptidoglycan cell wall overlain by an outer membrane containing lipopolysaccharides. Leptospira lipopolysaccharides are structurally and immunologically similar to LPs from other gram-negative organisms, although they do not appear to exhibit the same endotoxicity. Dark-field microscopy is used to visualize Leptospira, which are difficult to be visualized with conventional microscopy because they are so thin. Leptospira exhibit rapid darting movements in liquid media with flexing motions and rotation about their long axes. The characteristic motility of Leptospira is due to a periplasmic endoflagellum with polar insertions, which is unique to spirochete bacteria. The motility of Leptospira allows them to move in viscous environments, which enables them to penetrate blood vessels and rapidly reach target organs through hematogenous dissemination. Based on genetic typing, 22 species of Leptospira have been identified, which are divided into 3 distinct phylogenetic clades, see figure. The genus comprises a first clade of 10 pathogenic Leptospira species that have been associated with disease in humans and animals, a second clade of 5 species with intermediate pathogenicity traits that have been associated with mild clinical manifestations, and last clade of 6 saprophytic Leptospira species, which do not cause disease. Etiology. The smallest taxonomic unit of Leptospira is the serovar, and more than 250 distinct serovars have been described to date. Nomenclature for Leptospira serovars has changed over the years but now follows a format agreed by the Committee on the Taxonomy of Leptospira in 2002, where the genus and species are stated and italicized followed by the serovar name, which is capitalized but not italicized. This nomenclature is also useful as it reflects both the phylogenetic and serological classifications of each serovar. Although the serogroup is not strictly included in serovar nomenclature, it is commonly stated in parentheses after the serovar name. Cross-agglutination absorption tests and monoclonal and polyclonal agglutination tests are used to characterize serovars based on their serological profile, determined by structural heterogeneity in the carbohydrate component of the LPs. Related serovars are grouped into serogroups based on their serological and antigenic characteristics. The serological classification of Leptospira is complicated by horizontal transfer of the LP's biosynthetic locus, RFB, between different Leptospira species, resulting in a single serogroup being represented by multiple Leptospira species. Despite this limitation, serogroups remain essential for leptospirosis diagnosis and have proved useful for understanding population-level trends in predominant serogroups. Leptospira are fastidious organisms and may take up to 6 months to grow in vitro. Leptospira in culture grow at an optimal temperature of 29 to 30 degrees Celsius. Cultivation requires the supplementation of growth media with long-chain fatty acids and with vitamins B1 and B12. Fatty acids are supplied by the addition of serum to growth media, or alternatively, an albumin polysorbate medium can be used. Commercially available culture media include Ellinghaus and McCulloch-Johnson-Harris, EMJH, medium and Fletcher's medium, although some strains also require the addition of either pyruvate or rabbit serum. Protein-free media containing charcoal detoxified polysorbates are also popular, especially for vaccine production. Liquid, semi-solid or solid media can be used for cultivation. Most laboratories employ semi-solid media for initial isolation and maintenance of cultures and liquid media for antigen production. For isolation of Leptospira from non-laboratory sources, it is essential to add 5-fluorouracil or antibiotics in order to inhibit the growth of contaminating bacteria that may overgrow the target organism and hamper isolation. For clinical samples, it is essential to use fresh specimens of tissue or body fluids for isolation in order to lessen the detrimental effects of other bacteria and products of autolysis on Leptospira. Tissue and urine samples may also have to be diluted to minimize these effects and increase the chance of successful isolation. All mammalian livestock species can suffer Leptospira infections following contact with urine from an infected animal host or through sexual transmission. Contact with contaminated environmental sources such as soil, water or waterborne wastes such as slurry or runoff water from infected animal houses is epidemiologically important, as some Leptospira species can survive for prolonged periods in the environment. Favorable conditions such as adequate moisture and a neutral to slightly alkaline pH promote environmental survival. However, environmental persistence can vary amongst pathogenic Leptospira species. Certain species, such as Leptospira burgdorferi, appear poorly adapted to survive in the environment, while others, such as Leptospira interrogans, show prolonged environmental survival. A recently recognized factor that may influence the survival of Leptospira in the environment is their ability to form biofilms. This ability may be one of the main factors contributing to environmental persistence and disease transmission. Leptospira infection in livestock can be broadly divided into 2 major epidemiological categories. Infection with host-adapted serovars, such as L. burgdorferi serovar hardo in cattle, is characterized by subclinical infection with prolonged urinary shedding of bacteria. Animals that are chronically infected with host-adapted serovars are referred to as maintenance, reservoir or carrier hosts and are an important source of infection to other animals and humans. The second epidemiological category, described as incidental infections, includes infections of livestock with serovars that are adapted to and maintained by other animal species. Typically, these infections are associated with more severe clinical manifestations and may present as an outbreak of disease in affected herds. For example, L. interrogans serovar icterohemorrhagiae, a serovar that is carried and shed by brown rats, Rattus norvegicus, with few clinical signs of disease, can give severe disease and livestock infections, may be fatal. A wide range of mammal species may act as carrier hosts for pathogenic leptospira bacteria, with rodents considered a common source of leptospira serovars associated with incidental infection. Leptospira infection has also been detected in a range of other wildlife species by serology. However, the role of mammalian wildlife species in the epidemiology of leptospirosis in livestock is not well understood and warrants further research, particularly in regions where contact between wildlife and livestock is common. Leptospira antibodies have been demonstrated in non-mammalian vertebrates, fishes, reptiles and amphibians, and infection has been induced experimentally in chicken embryos. However, there is currently no evidence that non-mammalian hosts are susceptible to clinical disease or able to maintain and propagate leptospira infection, and therefore only mammalian hosts are thought to contribute to the epidemiology of the disease. Particular associations have been described between specific leptospira serovars and certain host species. Classic examples include L. interrogans serovar canicola in dogs, L. interrogans serovar pomona in pigs, L. Borg-Petersonii serovar hardho in cattle, and L. interrogans serovars copenhagenii and icterohemorrhagiae in rats. However, these associations are not absolute and may vary in different geographic settings. Of all livestock species, cattle appear to be the most susceptible to leptospirosis and are frequently implicated as the species with the highest seroprevalence. Where culture has been attempted, a wide range of serogroups have been isolated from cattle, although not all have been associated with clinical disease. Leptospira Borg-Petersonii serovar hardho, serogroup Sehro, a serovar that is associated with cattle worldwide, is known to have a reduced ability to survive in the environment. Infection with serovar hardho is therefore influenced more strongly by management systems that facilitate direct host-to-host transmission than environmental conditions, although environmental transmission may be possible in tropical conditions. For example, in Brazil, intensively managed dairy cattle were determined to be 8 times more likely to be infected with leptospirosis than extensively managed beef cattle from the same farm. In Africa, a serological survey of cattle at slaughter, where serogroup Sehro was the predominant reactive serogroup, showed a high seroprevalence even in dry areas, supporting the disconnect between environmental conditions and transmission of this serovar. The serovars most commonly associated with pigs are L. interrogans serovar pomona, serogroup Pomona, and L. interrogans serovar bratislava, serogroup Australis. Venereal transmission is thought to play an important role in the transmission of Bratislava amongst pig herds, which has been isolated from the genital tracts of sows and boars. Lack of rodent control and the use of artificial insemination have also been implicated as risk factors for leptospirosis in pigs. In temperate climates, the risk of infection in pigs has been shown to be related to rainfall, suggesting a potential environmental component to transmission. Evidence that pigs may act as reservoirs for leptospirosis appears to vary. A number of surveys have demonstrated a relatively low seroprevalence amongst pigs compared to other livestock species. In Hawaii, however, an increase in the incidence of disease caused by Serovar australis has been postulated to be due to increasing populations of feral pigs and their movement into urban areas, and, in Australia, feral pigs have also been implicated in leptospirosis transmission. In Africa is widespread, with Serovars canicola, icterohemorrhagiae and pomona isolated from pigs in infected herds. Sheep and goats have typically been thought to be incidental hosts of leptospira and not considered important reservoirs of infection, but recent studies have demonstrated that both species may act as carriers of pathogenic leptospira bacteria. Urinary shedding in sheep has been demonstrated in an abattoir study, and sheep have, along with cattle, been implicated as maintenance hosts of Serovar hardho. Infection in goats has also been reported in association with chronic renal and genital tract carriage, reproductive problems and abortion outbreaks. Although sheep and goats have been thought of as the least susceptible of domestic animals to leptospirosis, severe outbreaks may occur. Horses are usually implicated as incidental hosts of leptospira, although leptospiral DNA has been detected in the urine of horses following infection, suggesting a possible role as a source of infection for humans or other animals. Moreover, demonstration of leptospira DNA in equine semen suggests a potential venereal route of transmission in horses. Infection of horses with leptospirosis has been reported from Australia, North America, Egypt, and the UK. Pathogenesis. Leptospira infection is acquired through direct contact with urine or indirectly by water or mud contaminated with infected urine. Leptospira bacteria penetrate the mucous membranes or abraded skin and enter the bloodstream of the host, where they multiply and establish a leptospiremia. Thereafter, organisms localize to target organs such as the kidneys, lungs, brain, eyes and genital tract of both male and female animals. Leptospira have also been isolated from the cerebrospinal fluid of infected animals. Dissemination of leptospira is facilitated by hematogenous spread to tissue sites and is aided by the ability of pathogenic leptospira to cross tissue barriers, migrate through the extracellular matrix and adhere to host cells. Hemorrhage and disruption of hemostasis are also common manifestations of acute leptospirosis. The pathogenesis is still not fully understood but includes endothelial damage, disruption of tissue integrity through the production of bacterial toxins or enzymes such as sphingomyelinases. Leptospira has been shown to bind fibrinogen, plasminogen and partially inhibit fibrin formation, all of which may also contribute to reduced clotting ability. Furthermore, some Leptospira cerevars produce a hemolysin that causes hemolytic anemia and subsequent hemoglobinuria and icterus. Intravascular hemolysis can also contribute to liver damage and jaundice through ischemic injury to hepatic centrilobular areas. Secondary clinical signs, seen after an incubation period of 2 to 16 days, are dependent on the site of leptospiral localization. Localization in the proximal tubules of the kidneys in maintenance host causes mild chronic interstitial nephritis and long-term leptospiruria. Endothelial damage and subsequent ischemia are thought to be important mechanisms in tissue damage associated with leptospira infection. Enzymatic damage, from sphingomyelinases, to tissues may also contribute to the pathology associated with acute disease. Acute renal dysfunction is thought to be a combined result of reduced renal perfusion and ischemia as a consequence of intravascular hemolysis, combined with direct tissue damage and inflammation following leptospiral renal invasion. Chronic renal injury is associated with an inflammatory response to invading leptospira organisms, resulting in a lymphoplasmacytic infiltrate in the renal interstitium. Fetal infection, caused by leptospira crossing the placenta, varies in outcome depending on the age of the fetus at the time of infection. Possible outcomes include abortion or stillbirth, fetal antibody production and clearance of infection, or poor neonatal viability associated with latent infections. Leptospira localized in the fetal brain may persist for long periods, giving rise to neurological signs in some cases. The pathogenesis of uveitis in horses results from intraocular leptospiral infection and an associated immune-mediated response. There is a close antigenic relationship between certain leptospiral antigens and lens and retinal proteins of horses that appears to aid in the development of chronic inflammation in infected horses. Chronic lymphoplasmacytic inflammation of the uvea can lead to irreversible ocular damage, including the formation of posterior synechii, increased intraocular pressure and retinal detachment. The clinical signs of leptospirosis in livestock vary with the affected species and the infecting cerevar. In general, the clinical signs seen in association with leptospira infection can be grouped into 4 main syndromes. 1. Severe, peracute disease with a high fatality rate, most commonly seen in susceptible young stock. 2. Acute febrile disease in adult animals. 3. Reproductive disease characterized by abortion, stillbirth and reduced fertility, and 4. Subclinical, chronic renal infection. Other disease manifestations may also be seen, such as recurrent uveitis in horses and, occasionally, meningitis in young stock. Although the clinical signs may vary by the organ system affected, in general there are a number of similarities between different livestock species. Pyrexia, acute renal failure, jaundice and hemorrhage are typical manifestations of acute and peracute leptospirosis. The development of an acute febrile reaction coincides with the leptospiremic phase of infection and is one of the earliest clinical signs of leptospira infection. Young stock are particularly susceptible to severe, acute infection. Reproductive disease characterized by abortion, stillbirths and neonatal deaths is common amongst livestock species. Abortion storms may be seen in groups of naive, reproductively active animals. However, subclinical chronic infection is also common in livestock and may only be detected in routine screening or investigation of poor reproductive performance and infertility. Leptospiroria occurs in acute, subclinical and chronic infections, but the duration of shedding may vary. Acute infection with a non-host adapted serovar may persist for 2 to 4 weeks post-infection, whereas shedding can continue for more than 2 years in chronic, subclinical infections with adapted serovars. Amongst livestock species, cattle appear relatively susceptible to acute disease following Leptospira infection. Globally, serovars from a wide variety of serogroups have been reported in association with clinical infection in cattle. Acute leptospirosis is characterized by fever, anorexia, malaise, hemoglobinuria, icterus and anemia in cattle. Affected calves may also have diarrhea. Fever may last from 1 to 5 days, and in experimental studies it has been reported that relapses recur for up to 53 days in 80% of infected animals. Severely affected calves die within 1 to 7 days of developing clinical signs. In adult animals, Leptospira infection is not usually fatal but may result in chronic renal infection, which is associated with mild, focal, interstitial nephritis. Agalaxia, also referred to as milk drop or flabby bag syndrome, is a feature of acute leptospirosis in adult lactating animals and has been reported in association with a variety of serogroups including Pomona and Hardho infection. Infection may result in a precipitous drop in milk production. All quarters of the udder may become flaccid, and milk may vary from thick, pasty and cream-colored to bloody with a normal consistency. These cases may test positive on the California milk cell test. This agalaxia resolves within a week and its course is not affected by treatment. It appears not to be a true mastitis as the affected udder lacks an inflammatory response, though leptospires can be isolated from the milk and udder tissue. Abortions and reproductive failure are also common presentations of Leptospira infection in cattle. In pregnant females, abortions may occur in the last trimester and occur from 1 to 12 weeks following acute infection. Other reproductive problems such as stillbirths or the birth of weak or premature calves may also occur in association with Leptospira infection, depending on the stage of gestation when infection occurred. Although numerous serovars have been identified as causes of leptospirosis in cattle, serovars of the Safro serogroup, in particular the Hardho serovars, have been recognized worldwide as being an important cause of bovine abortion. Infertility is also a feature of infection with Hardho in cattle herds, and low conception rates are reported in Hardho infected herds. Hardho infection has also been associated with early fetal loss, so may present as an early return to estrus in breeding animals, with no visible signs of embryo loss. It is also worth noting that there is a relatively poor correlation between serological status and chronic renal infection and shedding in cattle, leptospiroria that can persist for up to 450 days or longer. The duration of antibody response is variable and may decline to low or undetectable levels before infection is cleared from kidney or genital tract. Leptospirosis in pigs is an important cause of productivity loss in farming systems worldwide. Infection manifests most commonly as a reproductive syndrome characterized by abortions, infertility and the birth of premature and weak piglets. Abortion may occur from as early as 6 weeks gestation, although it mostly occurs towards term. Many litters reach term and are either all stillborn or are a mixture of stillborn and live, weak piglets. The most common serogroups associated with reproductive failure in pigs are Pomona, especially Leptospira interrogan serovars Pomona and Kennewickii, Australis, L. interrogan serovar Bratislava, and serogroup Tarasovi. Acute systemic disease characterized by anorexia, jaundice and renal failure can also be seen, especially in young stock, and is typically caused by incidental infection with a variety of serogroups including Canicola, Hebdomatis, Icterohemorrhagiae, and autumnalis. Asymptomatic chronic interstitial nephritis may be identified at slaughter in Pomona infections, white spot kidneys. L. interrogan serovar Canicola has also been isolated from breeding sows affected by abortion, poor neonatal survival and infertility. In general, sheep are thought to be relatively resistant to acute clinical leptospirosis. Acute systemic disease, characterized by icterus, hemolytic anemia, hemoglobinuria, fever and death, has been reported in lambs in association with infections by the Pomona, hebdomatis, Balam and Grippetyposa serogroups. This fatal hemolytic disease may kill up to 5% of the lamb crop, losses being experienced in 2 to 4-month-old lambs. 161. Fatal hemolytic disease accompanied by icterus and hemoglobinuria has been induced in experimental infection of sheep with L. interrogans serovar pomona. Abortion, stillbirths, and the birth of weak lambs have all been reported and associated with heart-o infection. Infection with other serogroups such as hebdomatis, australis and pomona are also thought to cause abortion, with rates of up to 20% being recorded. Ewes can also show total or only partial agalaxia, which resolves without treatment within 3 to 4 days. This syndrome may only be detected when lambs with a normal birth weight die of malnutrition. Goats are also susceptible to infection with leptospira. Severe disease is most commonly seen in young kids and is associated with incidental infection. In Brazil, outbreaks of abortion and neonatal death associated with leptospira infection have been reported. Infection with serovars from the Grippetyposa serogroup have been experimentally shown to induce an acute disease characterized by hemoglobinuria, anemia and icterus, and death in severe cases, whereas experimental infection with serovars heart-o and pomona caused only mild clinical signs including mild fever, and a drop in milk production in some animals. It is possible, however, given reports from other geographic regions, that leptospirosis remains an undiagnosed cause of production losses in small ruminant rearing systems. Most cases of leptospirosis in horses are subclinical, although, as with young of other species, foals are more susceptible to acute infections, which may be fatal. More common clinical syndromes include abortions, stillbirth, premature live birth and recurrent uveitis. Serogroups including australis, Grippetyposa, hebdomatis, icterohemorrhagia and pomona have been isolated from aborted equine fetuses. Chronic leptospiral infections in horses are more commonly associated with the development of recurrent uveitis, also referred to as periodic ophthalmia or moon blindness. Signs of uveitis develop sometimes years, after the initial infection, and manifest as painful keratitis or ophthalmitis. It is recurrent, uni or bilateral and characterized clinically by miosis, congestion of the sclera, photophobia, severe lacrimation and sometimes blindness. Although recurrent uveitis is an important cause of equine vision loss around the world, lesions. The gross presentation of leptospira infection varies with the respective syndromes in affected animals. However, many features are consistent among the major livestock species. In acute systemic disease, the macroscopic lesions are characteristic of a hemolytic crisis and septicemia, and include icterus, anemia, splenomegaly and subcutaneous and mucosal hemorrhages. Pulmonary edema, hepatomegaly and nephritis are also typical of acute cases of leptospirosis. Localization of leptospira in the kidney is associated with interstitial nephritis and tubular necrosis, which may result in the kidneys macroscopically appearing swollen and hemorrhagic. In subacute or chronic renal infection, multifocal white spots with or without a hyperemic border may be seen, which may be as small as 1 to 2 mm in diameter, and may be restricted to the cortex or also extend into the medulla. These lesions are sometimes accompanied by a few hemorrhages and small pitted areas. The renal capsule is adherent to the cortex over the affected areas. Histologically there is mild, focally disseminated, interstitial, lymphoplasmacytic nephritis. The infiltrate in the affected kidneys occurs primarily in the interstitial tissue and around the glomeruli, and while it consists mostly of lymphocytes and plasma cells, a few neutrophils and eosinophils may also be seen. Other lesions in the kidneys include degeneration, necrosis and atrophy of the tubular epithelium, few cellular and hyaline casts, and the development of fibrosis. Glomeruli may show swelling of the tuft, an accumulation of protein in the urinary space, and fibrosis of Bowman's capsule. Focal areas of calcification may occur in the cortex and in the medulla. However, leptospiral organisms are not necessarily associated with lesions in the kidneys. The presence of leptospira in the kidneys does not necessarily induce nephritis, and there is often no correlation between the severity of the renal lesions and the isolation of leptospira from the respective tissues, especially with host-adapted serovars. Few specific lesions occur in the placenta of animals that have aborted. The intercotyledonary tissue may be edematous, while the cotyledons may be fawn-colored and atonic. Hydropic degeneration and focal necrosis of the cotyledons occur in the affected placentas. The degree of change varies amongst animals and also between the cotyledons of the same animal. Leptospires may be detected histologically in the tissues of affected animals by the application of immunofluorescence techniques. Traditionally, silver stains and modified Werthenstari stains have been used in histopathological diagnosis. However, these stains are insensitive and have also been associated with false positive results. In systemic leptospira infection in juvenile livestock, the organisms may be demonstrated in various tissues including the kidneys, liver and lungs, and organisms can also be seen in the kidneys, placenta, cotyledons, and the liver of animals with subclinical infections. The pathology of leptospirosis in cattle varies according to the respective syndrome. In acute cases of systemic infection with an incidental serovar, pathology is consistent with that seen in other species as described above. In abortions, aborted fetuses may be quite fresh, edematous or autolyzed at the time of abortion. Leptospira organisms may be detected in the kidneys, and focal tubular necrosis, sometimes associated with the early stages of interstitial nephritis, may also occur. Few lesions are seen in weak, live-born calves, but kidneys may be edematous and hemorrhages may occasionally occur in the urinary bladder, lungs and kidneys. Abortions, which occur late in gestation, are often the most visible sign of leptospira infection in pig herds. In aborted fetuses, multifocal hemorrhages are the most common macroscopic lesions. In infected neonates, icterus is also a common gross finding. Histologically, acute hepatitis, lymphocytic myocarditis and interstitial nephritis are seen in these cases. Chronic interstitial nephritis is frequently found in slaughter pigs infected with leptospires. These lesions are seen as grayish foci, a few millimeters to a centimeter in diameter, in the cortex and medulla. Advanced lesions will suffer fibroses. Histologically, the lesions in the kidneys vary according to the stage of their development. Acute lesions are characterized by hyperemia, edema and degeneration of the tubular epithelium, but when infection becomes subacute, histology is characterized by the presence of monocytes and macrophages that are replaced at a later stage by plasma cells and lymphocytes. Even serovars of low virulence cause lesions in the kidneys. Leptospires occur in affected kidneys at all the stages of the development of the lesions. Initially they are present in all the structures, including blood vessels, but as the lesions progress in age they are restricted to the microvilli of the renal epithelium. Few lesions are seen in adult sheep with leptospirosis. Typically, the only macroscopic lesions are acute nephritis and concurrent hemorrhage, although hepatitis has also been reported. Abortion may occur in some cases, and leptospires have been detected by immunoperoxidase in the tissues of aborted or stillborn fetuses. In lambs and kids, systemic infection can cause a fatal hemolytic syndrome characterized by severe icterus, anemia, hemoglobinuria, and pale brown 168 to dark red or blackish kidneys. On cut surface the kidneys manifest dark brown streaks and the focal white spots indicating interstitial nephritis. Cell swelling and necrosis of few hepatocytes are seen histologically in these animals, while hemosiderin pigmentation of the tubular epithelium and clumps of leptospires, hyaline casts, hemoglobin and cellular debris also occur in the kidneys. The sequestration of leptospires in the horse's eyes causes progressive uveitis, thought to be the consequence of an immune-mediated hypersensitivity reaction. The anterior uveitis that is seen initially progresses during repeated attacks to involve the posterior uvea, cornea, retina and optic nerve, with resulting blindness. Lesions are rarely encountered in aborted fetuses, even though the infection may be diagnosed by immunofluorescence, which is most successful in the kidneys and the liver, or by isolation of the organism. Few lesions are seen on histology of aborted fetuses. In rare cases, dissociation of the hepatocytes, necrosis of individual hepatocytes, and confluent and multiple areas of necrosis can be seen. Nonspecific lesions include degeneration of the renal tubules and perivascular hemorrhages in the brain. Concurrent infections such as equid herpes virus 1 cause more severe lesions in the affected organs. In these cases, viral inclusions are present in the affected cells and serve to confirm the diagnosis. Diagnosis. The history and clinical signs in affected animals may indicate leptospirosis. However, due to the variable nature of the clinical signs, the diagnosis should be confirmed by laboratory tests. The slow-growing and fastidious nature of Leptospira makes isolation of little use as a diagnostic test, and therefore definitive diagnosis is achieved through serological or molecular detection assays. Typically, the incubation period for leptospirosis is 7 to 12 days, although it may range from 2 to 30 days. The first phase of leptospirosis is the leptospiremic phase, during which leptospires can be detected in the blood for up to 7 days. The second phase of the disease is characterized by an immune response during which antibodies increase from the second week after infection, which coincides with tissue localization of the leptospires and the onset of leptospiruria. Serological diagnostics. The two most commonly used serological tests for leptospirosis are commercially available enzyme-linked immunosorbent assays, ELISA, or the microscopic agglutination test, MAT. Although ELISA has been used with success in the diagnosis of leptospirosis in some species, particularly in differentiating between acute and chronic infections, it cannot be used for conclusive serological diagnosis due to its poor sensitivity and specificity, and requires confirmation with MAT or PCR. The microscopic agglutination test, MAT, is the gold standard serological test and can be used in any livestock species. The MAT requires maintenance of a panel of live Leptospira isolates, and the inclusion of local isolates in MAT antigen panels is critical and can help improve the sensitivity of Leptospira detection. A major limitation of the MAT is that a single MAT test performed on an acute blood sample from an animal may be insufficient for the routine diagnosis of leptospirosis in individual animals. Serology is a poor diagnostic tool for confirming the role of leptospires in specific cases of recurrent uveitis in horses. Positive serological titers are more common than the occurrence of uveitis, and many serologically positive horses are clinically normal. Antibodies, however, appear also to be synthesized intraocularly, particularly during the active stages of recurrent uveitis, as their levels in the aqueous humor may exceed those in the serum. Antibody levels in aqueous humor, obtained by paracentesis, that are in excess of those in the blood, or the detection of Leptospira antigen using PCR, are considered to be diagnostic for recurrent uveitis. Molecular diagnostics. Polymerase chain reactions can be used for detection of Leptospira infections during the leptospiremic phase of the disease or to demonstrate urinary shedding in chronic infections. PCR is a useful diagnostic tool in outbreak situations where a rapid diagnosis is needed. Moreover, sequencing of PCR products may provide valuable phylogenetic data. For example, sequencing of PCR products obtained directly from urine has demonstrated the presence in cattle of Leptospira that have not yet been cultured. Although PCR is currently the best diagnostic technique available for a rapid diagnosis of leptospirosis, it does have limitations. Some PCR techniques detect saprophytic Leptospira in environmental contamination, which would lead to false positives. Differential diagnosis. In cattle, the hemolytic syndrome caused by leptospirosis should be differentiated from other causes of a hemolytic anemia such as babesiosis and anaplasmosis. Other causes of abortion, stillbirths, neonatal deaths and/or infertility in cattle include brucellosis, campylobacteriosis, chlamydiosis, cocciolosis Q fever, Neospora infection and trichomoniasis, as well as a variety of viral infections such as bovine viral diarrhea, BVD, Rift Valley fever, RVF, blue tongue or infectious bovine rhinotracheitis, IBR. In pigs, the abortions, stillbirths and mummification caused by porcine parvovirus and other viral infections can be confused with leptospirosis. The multifocal, subacute to chronic nephritis in cases of leptospiral infection in pigs is similar to that caused by other bacteria such as Escherichia coli, Streptococcus spp, and Arcanobacterium pyogenes, and by the mycotoxins, ochratoxin A, citrinin and others, produced by Aspergillus and Penicillium spp. Control. The complex multi-host epidemiology of leptospirosis can make control of the disease challenging. In animals, a variety of antibiotics have been used to treat acute leptospirosis. Currently, a combination of penicillin and streptomycin is the treatment of choice. However, ampicillin, amoxicillin, tetracyclines, tulathromycin and third-generation cephalosporins have also been used. Antibiotics may differ in their ability to clear renal carriage of leptospira infection in chronically infected animals. For example, in hamster models, ampicillin was unable to clear the infection from the kidneys and heart, while doxycycline cleared the infection from all organs in 2 days. In dogs, treatment with doxycycline has been suggested for clearance of the leptospires from renal tubules. Antibiotic treatment may also play a role in the control of leptospirosis. For example, systemic treatment of herds may be considered in the case of abortion storms. New introductions to a herd should be treated to prevent introduction of the disease, and semen diluent should be treated with a combination of penicillin and streptomycin to kill any leptospires. The most appropriate antibiotic treatment or control regime will vary depending on the livestock species involved, management system and other considerations such as the withdrawal period for meat or milk products. Vaccination is a practical means of preventing clinical leptospirosis in many livestock species, but may only be partially effective due to the serovar-specific nature of vaccine-induced immunity and the presence of local serovars not included in the vaccine. A wide range of vaccines are available for use in farm animals, including several multivalent preparations. However, vaccination does not necessarily prevent renal colonization and leptospiruria despite conferring protection against clinical disease and abortion. The recommendations of the vaccine manufacturer should be followed as they differ for the different vaccines, but some general guidelines for effective vaccination can be given. A critical aspect of any vaccination program should be the establishment of protective immunity prior to breeding, as vaccination is unlikely to be effective if placental infection has already occurred. Pigs inoculated for the first time should receive 2 injections 4 to 6 weeks apart. Breeding sows should be vaccinated 6 weeks before breeding and revaccinated 4 to 6 weeks later. A booster should be given 4 weeks before each subsequent farrowing. Boars should be vaccinated every 6 months. Colostrum antibodies from vaccinated sows protect piglets from leptospiral infection for approximately 3 months. Piglets of vaccinated sows should therefore be vaccinated at the age of 3 months, when passive immunity is waning. Inoculation of piglets less than 1 month old has been shown to be ineffective. Previously unvaccinated cattle should receive 2 inoculations 1 month apart. Testing for urinary shedding with subsequent antibiotic treatment of shedders may also be indicated prior to vaccination in herds that have been diagnosed with leptospira infection. An annual booster is recommended by most manufacturers, although it has been recommended that cattle should be vaccinated up to 3 times annually in order to prevent abortion and infertility. Due to a lack of cross-protective immunity between leptospira serovars, vaccinated livestock may still be susceptible to leptospira serovars not included in vaccines. Biosecurity to prevent new introductions of leptospira into the herd is also an important component of leptospirosis control. Many wild animal species can carry leptospira infection and act as a source of infection for livestock. In particular, rodents are an important source of a wide diversity of leptospira serovars, and incidental infection of livestock with rodent-associated leptospira serovars may cause outbreaks of acute disease. Rodent control appears to be of particular importance to the control of leptospirosis in piggeries. Rodent control should, therefore, form an important part of leptospirosis control strategies.